Good afternoon, everyone. Thank you for coming. It is my pleasure to give a talk of my research today. This is not only a talk, but it's also my oral defense, dissertation oral defense. So your attendance and your encouragement will make my day. And first of all, my name is Su Yifan, and my advisor is Dr. Huang Daoli Lao Shi. And the issue I'm going to talk today is the interplay between simulation and phosphorylation. This is a relatively big issue, and I have two topics that covers this. Before I start these two topics, let me first brief you through the backgrounds of simulation. Sumo pathway is a series of transferring process of the sumo protein from several sumo mechanism components to its target protein. And when a newly translated sumo protein, it is in an immature form. And this immature sumo is first converted from immature form converted to a mature form of sumo by the isopeptidase. And later on, this mature form sumo will be activated by the E1 activating enzyme through the conjugation of the thiol bond. And later, the sumo will pass over from the E1 enzyme to the UBC9 conjugation, which is conjugated with the thiol bond. And afterwards, with the help of the E3 ligase, UBC9 will transfer the sumo to the target protein through the isopeptide conjugation. In the end, sumo will be removed from its target by the isopeptidase. Now, like other post-translation modification, simulation also play many roles in biological functions. For instance, bioreplication, carcinogenesis, cell signaling, cell cycle, and transcription. And the two topics of my talk today covers we have relationship with transcription and cell cycle. Now, many reports also found that sumo modification cross-talks with other types of protein modification. For instance, acetylation, ubiquitination, it also sumoylates itself, and also phosphorylation. Like there are some examples shows that kinases and phosphatides can be sumoylated, and also sumo machinery components can be phosphoregulated, and also at the substrate level, its phosphorylation, its sumolation can be enhanced or inhibited by phosphorylation. So, why do we study phosphorylation and sumolation? First of all, this last study, and it's relatively new, and especially the crosstalk between the sumo components and the phosphorylation. Now, for just take UBC9 or E1 enzyme for example. In simulation, there's only one E1 and one UBC9, and don't you think that it is incredible for just one E1 or one UBC9 to regulate the whole global simulation? So I assume there must be some kind of extra energy which helps or pushes the regulation, function. So therefore, we are interested to having better insight to see the linkage between the phosphorylation and simulation machinery. So this will lead you all to my first topic. My first topic is phosphorylation of UBC9 by CDK1/2B enhances the simulation activity, and It is just recently published. And first of all, what we did was we used several different kinases and tested if these kinases will have an effect on the simulation activity. Okay, we used these kinases, the CDK1 cyclin B, CDK2 cyclin E, ERK1 and 2, PKA, and JAK kinase. Okay. If you look at the top panel, in the increasing concentration of the CDK1 cyclin B, you will see that the increased simulation activity in the concentration-dependent manner. It stands out from other kinases because if you look at other simulation activity by other kinases, it seems that the simulation activity is not affected by the rest of the kinase.
But I don't quite agree. If you look at ERK1 or ERK2, I thought they are also increasing. Or even PKA.
But if you compare the intensity, comparing these few kinases, my personal feelings is that CDK1 cyclin B stimulates greater than, has a greater stimulatory effect greater than other kinases. That's my personal feeling. So that's why we think that CDK1 cyclin B is one candidate that we can further our studies. Yeah. So the substrate material that we use is a short peptide of the human topoisomerase 1. And because we want to see if the crosstalk, whether the crosstalk between phosphorylation and simulation machineries, so we first just mutated the serines in a terminal part to alanine, just to make sure that the kinase may not target to the substrate. And the advantage of using this substrate is that it contains a known sumo recognition motif, this part, and it is easy to prepare, like it's easy to express and easy to purify, and also it's relatively stable. And just to for knowledge, sumo recognition motif consists of these four amino acids. The 5 stands for the hydrophobic residue, K is for lysine, X for any residues, and E is for glutamic acid. Now, the previous result indicates that CDK1 cyclin B stimulates the simulation activity. So let's briefly talk about the background of the CDK. Now, CDK plays an important role in ensuring the proper cell cycle regulation when it associates with specific cyclins in specific phase and for specific functions. Now, a brief introduction of this kinase. Before the cyclin B, before cell cycle goes into the mitosis, cyclin B is synthesized in G2 phase. And then cyclin B will associate with CDK1 before and triggers the mitosis. And this kinase complex phosphorylates more than 70 proteins and it regulates the chromosome condensation, with Golgi network fermentation, as well as nuclear envelope breakdown. Lastly, CDK1 will, this kinase is inactivated to exit mitosis through the degradation of cyclin B. Now, the indispensable role of simulation in the cell cycle, particularly mitosis, can be seen throughout different species. Like for instance, I show here in the lower eukaryotes fission yeast, it has the sumo homolog called PMT3. The disruption of this PMT3 will show a defect in mitotic structure and segregation error. Now for its sumo protease in the body is called ULP1, its mutant will cause a cell cycle delay as well as the chromosome segregation.